So we're going to start talking about vision. Vision's arguably the most complex of all the special senses, and so we're going to break this up into two videos. I will let you know that I'm not planning on talking about like nearsightedness or farsightedness or how that all works. We're really going to just focus on how a process called phototransduction occurs, and then we're going to talk about how you can distinguish different colors over these two videos. And so here we go. So we're going to just really dive into phototransduction. So in order to understand phototransduction, we need to understand how light passes through your eye. So it enters your cornea and goes through your pupil, ultimately through a lens where it's focused onto the retina, and the back of your retina is where these photoreceptors are located. The light will activate these photoreceptors, and depending on which photoreceptors are activated, that will ultimately lead to what colors you perceive. Okay, so when we talk about phototransduction, what we're talking about is we're taking light and we're converting that into an electrical signal. And then from there, that electrical signal is turned into a chemical signal, and it does this back and forth between electrical and chemical a couple times before ultimately we activate the optic nerve. This is why vision is complex, and it actually takes a relatively long period of time because we have to convert, or we're going through multiple cells and we're undergoing multiple stages of transduction. So in the retina, when it comes to phototransduction, we need to focus on, or we need to be concerned with three different types of cells. So you've got the photoreceptors. Photoreceptors, there are rods and there are cones. Okay, we will spend most of our time actually talking about photoreceptors in general, but we will focus on how we can sense color in the next video, and that's dealing specifically with cones. Now, rods are located on the periphery or the edges of your retina, and we use these when it's low light. And what I mean by that is when it's dark. Your cones, on the other hand, are located in the middle. These are used when there's high light or when it's bright outside. Then you've got bipolar cells. These are located in what we call the outer synaptic layer. And then you've got ganglion cells. These are in the inner synaptic layer, and ganglion cells ultimately communicate with the optic nerve. Okay, most of what we talk about with phototransduction will occur with these photoreceptors. And so, just a reminder, when it comes to sensory receptors, most sensory receptors do not have action potentials. Instead, they have graded potentials. Photoreceptors and bipolar cells don't have action potentials, but ganglion cells do. Okay, and so let's get into these photoreceptors. Now, in order for phototransduction to occur, it's a complex process that involves some proteins and enzymes. So you've got something called rhodopsin. Okay, rhodopsin is made up of two primary parts. You've got retinal and opsin. Now, rhodopsin is actually inactive in the dark. So rhodopsin becomes active when there's light. Then you've got something called transducin. Transducin is a G protein. Now, normally, because it's inactive, it has GDP attached to it. Remember, GDP inactivates G proteins. And you're going to see that this G protein pathway is similar, not exactly the same, but similar to the G alpha I pathway. And then you've got phosphodiesterase, or PDE. This is an inhibitory enzyme. And if you recall from our discussion about G proteins, the G alpha I pathway can activate PDE. And so we're activating something that inhibits other things. Now, just like all neurons and all cells, there is a resting membrane potential. Typical neuron resting membrane potential of about -70. That's a little bit different in photoreceptor. Okay, we have what are called cGMP gated channels. Okay, that just means cGMP is the key that opens up those channels, and when they're open, sodium and calcium are able to flow through. And we're going to focus primarily on sodium when we talk about these channels. There's potassium leak channels, which we've talked about. They allow potassium to leave photoreceptors. And there's the sodium-potassium ATPase pump, just like on a regular regular neuron. Ultimately, this causes a negative resting potential of about -40. So it's not as negative as a typical neuron, but it's still negative. And so here is a diagram that shows everything that we're going to talk about on the rest of this video. There is a lot going on here. Okay, but ultimately what's going to happen is we're actually going to inhibit the release of a neurotransmitter when there's light out. Okay, but we'll talk about this in just a minute. So how does this work? Well, light shows up. Focus on the retina, we activate these photoreceptors. Remember how we said that there's this protein called rhodopsin? Well, rhodopsin is activated by light, and when it's activated, it actually breaks apart. So we've now got retinal and opsin. We're not going to worry about retinal, we're going to focus on opsin. And so opsin is what actually activates the G protein. Okay, opsin will bind to the G protein. That causes GDP to leave and GTP to attach. Just like every other G protein pathway we've talked about, GTP and alpha leave and they bind to an enzyme or a protein. And in this case, that protein is PDE, that phosphodiesterase. Remember, PDE is inhibitory. And so PDE is activated. Okay, so we've got this active thing that is going to inhibit something. And we'll get to that in just a second. Okay, I want to bounce back real quick to the gates that we have on the photoreceptors. Remember I mentioned those cGMP gated sodium channels. Yes, calcium is involved, but we're focusing on sodium. Okay, so we call them cGMP gated channels because cGMP is the key to open them up. And so if I have cGMP, sodium influx occurs into the photoreceptors. That allows these photoreceptors to become depolarized. This is where vision gets complicated and a little unintuitive. I don't know if that's the right word. Okay. So what happens is sodium goes in, that depolarizes the photoreceptors, which causes glutamate to be released. Glutamate, in this case, inhibits most of those bipolar cells. We mentioned bipolar cells earlier. Okay, bipolar cells basically connect photoreceptors to ganglion cells. Well, glutamate, like I said, inhibits most bipolar cells, or in other words, it causes these bipolar cells to become hyperpolarized. And so they're not going to be as efficient as doing their job. The reason we call bipolar cells is that glutamate inhibits most bipolar cells, but it excites some bipolar cells. We're going to focus on the bipolar cells that are inhibited by glutamate. Another thing that makes this a little counterintuitive, that's the word I'm looking for, is that these cGMP gated channels are open when it is dark. Okay, glutamate is being released by photoreceptors when it is dark. So in other words, there's no stimulus, but yet we're releasing neurotransmitter. Remember, graded potentials. So this takes us back to PDE. Remember, we get PDE when it's light. Okay, well, PDE breaks cGMP into GMP. Basically, it's breaking the key that opens the door. If I give you a broken key, you can't open the door. So as a result, those photoreceptors become hyperpolarized because sodium can't enter into the cell. Remember, we have potassium leak channels, so potassium can still leak out. And so those two things in combination will hyperpolarize photoreceptors. When you hyperpolarize any neuron or any cell, it's going to release less neurotransmitter. Okay, so as a result, glutamate release is reduced. Okay, this allows bipolar cells to become depolarized. Remember, glutamate inhibits most bipolar cells. However, in this case, because we're releasing less glutamate, that allows bipolar cells to be depolarized. Again, counterintuitive. So in darkness, let's just go through this really quick. So there's nothing stimulating the photoreceptor, okay? And so as a result, the actual thing that senses light isn't activated, and so we don't have the splitting of rhodopsin, which means we have less PDE, shown here. And so as a result, these gates are open, so sodium as well as calcium can enter the cell, okay? As a result, photoreceptors, like it says here at the bottom, photoreceptors are depolarized, which allows for the release of more glutamate. This release of glutamate hyperpolarizes bipolar cells, and so bipolar cells don't do their job. If we come to this one, notice we've got the light. It activates the photoreceptor. Rhodopsin splits into retinal and opsin down here. Opsin comes to here with the G protein, that's transducin. GDP out, GTP in, and then GTP and alpha go to PDE. And so now we've got an active hammer. And so with that hammer, we break cGMP into GMP. As a result, notice that this gate is now closed because we don't have the key. We broke the key. Okay, there is still potassium leaving the cell, that's fine. So the photoreceptor becomes hyperpolarized, but what happens as a result, and we can see this here in this summary, is that less glutamate is released from the photoreceptor. Okay, so in other words, we're getting rid of the inhibition. Glutamate inhibits most bipolar cells. So if there's no glutamate, we get rid of that inhibition. Okay, and so as a result, bipolar cells can do their job. The way that I think about this, or one of the ways that I think about this, is let's just say that you want your significant, you're living at home, and you want your significant other to come over. Okay, well, your parents are the inhibition. Maybe you don't want your significant over when your parents are there, just because. Okay, they're the inhibition. But as soon as the parents leave, your significant other comes over, and you guys hang out or do whatever. Right? So we get rid of the inhibition. So in this case, glutamate is like your parents. We get rid of your parents. That inhibition is gone, and so now you and your significant other are able to hang out. So let's finish real quick talking about bipolar and ganglion cells. So bipolar cells can excite or inhibit, okay? And it depends on the specific bipolar cell, all right? It's not just right now it's exciting me, and then a little while later it's going to inhibit. It's a specific type of bipolar cells. So since light generally leads to depolarization of bipolar cells, okay, remember we get rid of that inhibition. What's really interesting is that these bipolar cells actually release glutamate. Okay, so it's inhibited by glutamate, but then it releases glutamate. All right, so again, it's a little bit of one of those funky things that happens in physiology. Well, bipolar cells will release glutamate, which communicate with ganglion cells. Bipolar cells have graded potential, so it will just change the amount of glutamate that they're releasing. So if they release more glutamate, that's ultimately going to activate ganglion cells. So ganglion cells are depolarized by this glutamate, and when they're depolarized, they release neurotransmitter. Ultimately, this causes the signal to be sent along the optic nerve, and you see something. And so these ganglion cells are activated when it's light. So when it's dark, photoreceptors are depolarized, so they release more glutamate. So your parents show up, so you don't want to hang out with your significant other or do whatever it is that you want to do when your parents aren't around. Okay, so as a result, this glutamate inactivates most bipolar cells. So they release less glutamate, which means the ganglion cells aren't activated because they're not getting the signal to do their job. And so your brain goes, nothing to see here. So when it's light, photoreceptors are hyperpolarized. They release less glutamate. This activates most bipolar cells. As a result, those bipolar cells release glutamate. This glutamate activates ganglion cells, sends a signal along the optic nerve, and your brain goes, hey, I see a computer screen, or I see my orange water bottle, or whatever it is I see. So at the end of this, can you identify the 3 types of cells of phototransduction, and can you summarize what's going on with phototransduction? I want to end by saying I know that this is complex. It's probably scary. Some of you are probably pulling out your hair right now. But just trust me, we can get through this, and we will get through this with the help of the UTAs, with me, and with your fellow classmates. You can absolutely do this.